We integrated all 5 omics layers: genomics, transcriptomics, proteomics, metabolomics, epigenomics, PCA on the full concatenated matrix. The tumor subtypes separate cleanly. 3 distinct clusters. The figure is already in the deck.
5 omics. That is a lot of omics.
3 clusters, 5 platforms, 1 platform licensing deal. I am calling this a multi-omics biomarker panel. Tumor view, Series A closes before the replication study.
Which layer drove the separation?
All of them. That's the definition of integration.
How many features did your transcriptomics layer have?
Around 20,000 gene expression features.
Your proteomics layer?
300 proteins, maybe 350.
Your metabolomics?
800 or so.
You concatenated those into a single matrix and ran PCA. PCA maximizes variance. Transcriptomics contributed 20,000 features. Everything else contributed 1,100. The first principal component is RNA-seq signal. Your clusters are transcriptomic batch clusters. The other 4 omics contributed approximately nothing to the separation.
We z-score normalized every layer before concatenation.
Z-score normalization scales the values within each feature. It does not change how many features each omic contributes. 20,000 normalized transcriptomic features still dominate the PCA. You did not integrate 5 omics. You ran a transcriptomics analysis with 8 weeks of proteomics and metabolomics data attached to the column names.
Wait, the integration was fake the whole time? The proteomics and metabolomics were literally passengers? They were there, and PCA just ignored them?
Their contribution to the principal components was proportional to their feature count divided by the total feature count, which was small. Yes.
Okay, but how wrong is this? Like, how far are the clusters from what they should be?
Unknowable without knowing the true subtype structure. The clusters you have describe transcriptomic variation. What the metabolomics would show independently is a different question you have not answered.
It is mildly instructive that the figure looks compelling regardless.
Can I call the clusters molecularly distinct?
No.
Can I call them transcriptomically distinct and mention the other omics as supporting context?
You could describe it accurately, yes.
I would prefer not to.
To understand what real multi-omics integration requires, we need to talk about what it means for multiple matrices measured on the same patients to share information, and why the math has to do more than stack the columns. Multi-omics integration is like trying to understand what a factory is doing from 3 separate files: the shipping manifest, the power consumption log, and the employee attendance record. Each file is real. None of them shows you what the factory makes directly. The thing you are trying to find, the disease subtype, the pathway state, the biological mechanism, is the factory. Each omic is one file. Concatenating the files and running PCA is reading only the shipping manifest because it is the longest file and ignoring the other 2. The factory is still there. You just did not look at it. If you want to find out what your factory is actually doing, subscribe. New episode every Saturday. Multi-omics data means you have measured the same biological system at multiple molecular layers simultaneously. Genomics gives you DNA-level variation, mutations, copy number alterations, structural rearrangements. Transcriptomics gives you RNA abundance, which genes are being read and at what level. Proteomics gives you protein abundances, what is actually being produced from the transcripts. Metabolomics gives you small molecule concentrations, the downstream output of cellular activity. Each layer is a different file describing the same factory. They are not the same kind of measurement. They do not have the same statistical properties. They do not have the same feature counts. And they often do not have the same samples.
Wait, they don't even have the same samples? You measured all of this from the same patients, right?
Not necessarily. Proteomics requires higher sample quality than RNA-seq. Metabolomics has its own extraction protocol with different failure rates. In a real cohort of 100 patients, you might have clean transcriptomics on 95, proteomics on 80, and metabolomics on 60. The 60 metabolomics patients are not a random subset. They are the patients with the best sample quality. That is not random.
So how do the methods handle the different feature counts, the different scales, the missing samples?
The serious methods do not combine the raw features at all. They find a shared representation across the matrices. There are 3 main approaches. They solve the problem differently, and they answer different questions. The first is factor-based integration. MOFA, multi-omics factor, Analysis uses a probabilistic latent factor model. Formally, for each omic m, you model the data as y sub m equals w sub m times z plus epsilon sub m. Y sub m is the data matrix for omic m, features by samples. Z is the latent factor matrix, k factors by n samples. W sub m is the loading matrix, features by k, specific to that omic. Epsilon is Gaussian noise. Z is shared across all omics. Every omic is trying to explain its own data using the same underlying factor matrix. A factor that explains variance in multiple omics simultaneously is a factor with cross-omic signal. A factor that loads only onto one omic is probably a technical artifact from that platform.
So if a biological signal only shows up in transcriptomics and proteomics but not in metabolomics, is that valid? Does MOFA just throw it away?
MOFA handles it through sparsity. Each factor has a prior that pushes its loadings toward 0 unless the data requires them to be non-zero. The model learns which omics each factor is active in. A factor active in transcriptomics and proteomics, but inactive in metabolomics is valid. It might represent post-transcriptional regulation that has not propagated downstream. The sparsity pattern is itself biologically informative. If a factor is dense across all 5 omics, check whether it is a batch effect, not a biological signal. MOFA is implemented in the MOFE2R package from Bioconductor. In Python, the same model is available through MOFE PI2 and is typically used within the muon ecosystem. You store each omic as an AND-data object, combine them into a mu-data object, and pass it to MOFA+. You specify the number of latent factors k, usually between 5 and 25, depending on the complexity of your dataset. The model fits by variational inference. The second approach is similarity network fusion. SNF does not look for latent factors at all. Instead, for each omic m, it builds a patient similarity network, an n by n matrix W sub m, where entry ij is how similar patient I and patient j are based on that omic alone. You then run an iterative diffusion update. Each network is updated using information from all other networks simultaneously. Formally, W sub m gets replaced by P times the average of the other networks times P transpose, where P is a normalized diffusion kernel derived from W sub m itself. You repeat until the networks converge. The fused network reflects patterns consistent across all omics. SNF tool in R and SNFPY in Python implement this.
But what if one omic has a really strong signal? Doesn't it just take over the fused network the same way transcriptomics took over the PCA?
SNF is more resistant to this than concatenation because the diffusion step normalizes the local structure of each network before fusing. The influence of each network depends on its local geometry, not its raw feature count. A dominant omic can still bias the result. It is not immune, but the iterative diffusion resists takeover in a way that column concatenation does not. It is not perfect. It is better. The third approach is supervised integration. Diablo, from the Mix-omics R package, finds linear combinations of features within each omic that jointly maximize covariance with a known outcome. A tumor subtype, a treatment response, a survival class. Formally, it extends sparse canonical correlation analysis to more than 2 datasets. For each omic m, it finds a weight vector, a sub m, such that a sub m transpose y sub m has maximal covariance with the outcome and with the projections from the other omics simultaneously. It is a discriminant method. It requires labels.
So Diablo only works if you already know the groups you are trying to find?
Yes. Diablo is for when you have a labeled outcome and want the multi-omic signature that predicts it. MOFA and SNF are unsupervised. They find structure without requiring labels. These are different questions. If you want to discover unknown subtypes, use MOFA or SNF. If you want to build a multi-omic predictor for a known clinical outcome, use Diablo. If you use Diablo to discover subtypes by treating cluster labels from a previous PCA as the outcome, you have run a circular analysis. A well-fitted MOFA result gives you K factors ranked by the variance they explain across omics. Factor 1 might explain 30% of transcriptomic variance and 22% of proteomic variance. That is a candidate biological signal. Factor 2 might explain 18% of transcriptomics and 50% of metabolomics, potentially a metabolic phenotype with a corresponding transcriptional signal. Factor 3 might explain 70% of its total variance from a single omic and near zero from the others. That is a platform-specific technical effect. Treat it as a covariate, not a biology. The factor weights tell you which features load onto each factor. If factor 1 has high weights on a coherent set of transcripts and the corresponding protein products also load heavily on factor 1, you have found a multi-omic signal with mechanistic coherence. If the high-weight transcripts load on factor 1 and the corresponding proteins load on a different factor, you have found a post-transcriptional regulatory event. The structure of the weight matrices is the finding. A broken multi-omics integration looks like the cold open. Factor 1 is transcriptomics. Factor 2 is another RNA-seq batch. The loading vectors for your proteomics and metabolomics in every factor are near zero. You ran transcriptomics. If you have been publishing PCA plots from concatenated multi-omics matrices, this is the specific check you need to run. For each principal component, compute the percentage of variance explained by each omics features. If one omic accounts for more than 70% of a component's variance, that component is not integrated. It is that omic. Missing data in multi-omics is not missing at random. The patients who completed all 5 omics platforms are systematically different from the patients who did not. They had better sample quality. They were more likely to have remained in the study at a later time point. They were more likely to be from the center with the better biobank infrastructure. Excluding incomplete cases does not give you a random subset. It gives you the best case subset. MOFA handles missing data by marginalizing over the missing entries during inference. You can fit the model on partially overlapping samples. SNF requires full overlap or imputation. Know which situation you are in before you proceed. Factor count selection in MOFA is not a free parameter. You can tune upward until the results look good. More factors means more noise patterns captured as signal. The correct approach is to fit with a generous K, and then discard factors that are active in only one omic, explain low total variance, or correlate with known technical covariates. Library size, sequencing depth, batch ID, sample processing date. If your biological factors survive that filter, they are candidates. If they do not, they were batch effects with interesting labels.
What is the minimum number of samples you actually need for this to work?
For MOFA to reliably distinguish K factors, you need roughly 10 to 20 times K samples per omic. If you are fitting 10 factors on 60 samples, you are in the regime where noise will produce spurious factors. The solution is more samples. There is no tuning parameter that compensates for inadequate N. Factor weights are not biomarkers. A high weight on a gene in factor 3 means that gene covaries with the other genes that load on factor 3, in these samples, in this dataset, on this platform. It does not mean the gene is causal. It does not mean it will replicate in an independent cohort. It does not mean it is clinically actionable. It is a correlational pattern in a specific matrix. The appropriate downstream step is hypothesis generation and experimental validation, not a press release. Multi-omics integration does not give you directionality. A correlated pattern between gene expression and protein abundance across a factor does not establish that the gene is driving the protein level. It establishes that they vary together across your samples. Causal direction requires either temporal data, perturbation data, or a causal inference framework applied after the integration step. Integration shows you that the signals are consistent. It does not show you why.
It is mildly instructive that the majority of published multi-omics subtyping results fail to replicate in independent cohorts. Within-cohort clustering appears stable. Cross-cohort stability is substantially lower. The gap is partially sample size. It is also partially that the integration was absorbing batch variation that does not transfer to a new sequencing facility.
Could we call the non-replicating factors exploratory?
You could describe them accurately, yes.
Could we describe them as hypothesis-generating multi-omic axes of variation?
That is what I said.
Great, the deck stays.
A 5-omic study on 100 patients is expensive. Not slightly expensive. Substantially more expensive than a single omic study on 500 patients. If your integration method produces factors dominated by the most feature-rich omic, you have spent the money and not used the data. The metabolomics, the proteomics, the epigenomics, they exist in your repository and they do not appear in your findings. The biological question that motivated the study, what is the shared molecular signal across layers, has not been answered. It is also mildly instructive that the platforms producing the most features have historically received the most analytical attention, which is exactly backwards if you believe that proteins and metabolites are mechanistically closer to the phenotype than transcripts. Transcriptomics dominates multi-omics analyses, not because the biology is there, but because the feature count is. The correct approach is not complicated. Build separate similarity matrices or fit separate factor models per omic, verify that the integration method genuinely combines them, confirm that each omic contributes to the shared factors, and then report which factors survive filtering for technical confounders. The method exists, the software exists, the obstacle is not computational. Even after you run the analysis correctly, you still do